Okay, good morning, everyone. Again, I'm Jave, and I'm a third-year PhD student in Nick's lab at the Sainsbury laboratory. So this morning, I'll be sharing with you an aspect of my PhD project, which focuses on the identification of a novel group of phosphorylated effectors in the rice blast fungus Magnaporthe oryzae. So I am studying this particular disease of rice called rice blast, and this is the most economically important disease of rice. So typical symptoms that you can see in the field, you can observe in the field is this grayish lesion with dark brown spot. And this particular disease is actually contribute to up to 30% annual yield loss worldwide, which is enough to feed up to 60 million people annually. And this disease is a consequence of a successful and rapid colonization of the fungal pathogen called Magnaporthe oryzae. So I'm showing you here in this video the cell-to-cell movement and invasive growth of the Magnaporthe oryzae. And this invasive growth and cell-to-cell movement of the pathogen is regulated by a single fungal MAP kinase called PMK1, or the pathogenicity mitogen-activated protein kinase 1. So PMK1 is such a massive regulator of pathogen infection, of blast pathogen infection, such that when you delete the PMK1 in the pathogen, it renders non-pathogenicity to the blast fungus, compared to the wild-type isolate G11, which is capable of generating this infection structure called apresorium. PMK1 is unable to develop that structure. And compared to the wild-type, it's able to induce lesions when inoculated on leaves. So to further characterize the function of PMK1 during the invasive growth stage of the pathogen, so an analog-sensitive mutant version of PMK1 was generated in the lab, and we call that as PMK1 analog-sensitive. And this is particularly sensitive to the action of a chemical inhibitor called naptyl-PP1. And in the absence of this chemical inhibitor, the PMK1-AS acts like a wild-type PMK1. You can see in here that the pathogen is happily growing inside the host living tissue. But in the presence of this chemical inhibitor, the PMK1 function is arrested, and therefore the pathogen is unable to move from one cell to another cell. So in order for us to determine what are the downstream targets of PMK1, what are the components during its phosphorylation during invasive growth stage, so we perform a comparative phosphoproteomics. In this assay, we use similar strain called the PMK1-AS and then inoculated it in rice leaf sheet, and treat it with or without the chemical inhibitor naptyl-PP1, and then subjected the infected rice materials to phosphoproteomics analysis like protein extraction and then phospho-enrichment and then mass spectrometry. And that allowed us to identify a total of 42 differentially phosphorylated proteins that have MAP kinase motif, and their functions or their phosphorylation is dependent when PMK1 is functional. So looking into those 42 phosphoproteins, so those proteins are involved in different processes, such as in cell division, cytoskeleton remodeling. Some are transcription factors, some are involved in protein synthesis, and some are involved in secretion or in vesicular trafficking. But a large portion of it are uncharacterized proteins. Surprisingly, when we check for those uncharacterized proteins, 7 of them are small proteins, and that they contain signal peptide. And signal peptide is a molecular signature that a protein can be secreted out of the cell. So in this cartoon, I'm showing you those 7 phosphorylated proteins with the signal peptide and the identified phosphoserine residues. And because their expression is dependent on PMK1, I term them as PMK1 regulated effectors. And we are reminded, for instance, that our lab recently reported that during invasive growth stage of the pathogen, it secretes a large battery of effector proteins, such as 548 predicted proteins. And because of this, we focus on characterizing these phosphorylated proteins. So now, for the sake of my presentation, I will just be discussing these three PREs, or PMK1 regulated effectors. Now, our big biological question then is, what is the role of PREs during invasive growth stage of Magnaporthe oryzae? So in Magnaporthe, effectors can be classified into apoplastic or cytoplasmic effectors depending on their localization once they are secreted by the fungus into the host cell. So they are apoplastic effectors if they are localizing in the apoplast of the host, And if they are classified as cytoplasmic effector if they localize in this structure called the biotrophic interfacial complex. And indeed, when I clone my PREs fused to M. cherry and express that in Magna Porthae oryzae, after 30 hours post-infection, I'm seeing them localizing at that structure. They are preferentially accumulating at that structure called the biotrophic interfacial complex, or BIC, indicating that they are secreted proteins and that they are cytoplasmic effectors. And to further demonstrate that PREs 1 to 3 can be delivered into the host cell, I clone PREs and then fuse it to GFP with nuclear localization signal. And indeed, so it preferentially accumulates at the biotrophic interfacial complex, and at the same time, it also accumulates in the host nucleus, indicating that they are delivered and being uptaken by the host. So this cell biology assay validated that PREs 1 to 3 are secreted proteins and are cytoplasmic effectors. Because effectors are involved in promoting susceptibility and in suppressing host immunity, so I then asked the next question, are they involved in the virulence of the pathogen? So I then decided to knock out PREs 1 to 3 by CRISPR-Cas9, and compared to G11, null mutants of PRE2 produce smaller lesions compared to the wild-type G11, as you can see in this infection assay. But that is not that wasn't the case for knockout PRE1. And for my PRE3, the phenotyping is still in progress. So indeed, PRE2 is involved in is required for the full virulence of Magna Porthae oryzae pathogen. And because we identified PREs as phosphorylated effectors from our infected device phosphoproteomics analysis, we would like to determine where exactly they are phosphorylated. Are they phosphorylated before they are secreted by the pathogen, or are they phosphorylated in planta? To answer that question, so I allowed my PREs to be expressed in the absence of the host by performing transient expression in Nicotiana bentamiana, and then after 48 hours of agroinfiltration, I then perform protein extraction and then immunoprecipitation to capture my PREs and then subject the materials for mass spectrometry. And our mass spectrometry revealed that PREs are also phosphorylated in planta at the same serine residues that are found from our rice proteomics, indicating that PREs are phosphorylated in planta. So in summary, so our lab previously demonstrated that PMK1 is a mass regulator of Magna Porthae invasive growth stage, and that PREs at the same time is regulating as a subset or a novel group of phosphorylated effectors, which I call as PREs or PMK1 regulated effectors. PREs 1 to 3 are cytoplasmic effectors, are secreted proteins, and that deleting PRE2 impairs the pathogenicity or the virulence of the blast fungus, and that phosphorylation can happen in planta. So our current working model now is that during invasive growth stage, the pathogen, which is controlled by PMK1, the pathogen secretes these 3 cytoplasmic effectors, and they are phosphorylated in planta by a host kinase, MAP kinase, or MAP kinases, which we still do not know yet. But our outstanding questions, which we want to address in the future, is what exactly is the role of phosphorylation in the virulence function of these effectors, and what are ultimately we'd like to identify their host protein targets to really elucidate their virulence mechanism. So with that, I would like to thank my supervisory committee members, my primary supervisor, Professor Nick Talbot, the members of the Talbot Lab, and my collaborators, members of the proteomics team, and different support teams from TSL and JIC, and my scholarship, Hopkins Scholarship for Rice Blast Research. And thank you all for your attention.